The production of this program is made possible thanks to the support of the following and viewers like you.
Do you know your A's, C's, G's, and T's? They are the 4 types of bases in a DNA molecule, which consists of 2 strands wound around each other. An organism's complete set of DNA is called its genome. DNA carries the instructions for making specific proteins or sets of proteins. There are about 20,000 genes in the human genome, located on 23 pairs of chromosomes, which are packed into the nucleus of the human cell. It's remarkably complex and remarkably important as we move towards a future that will allow us to make informed and specific decisions about our health, the health of all other living beings, plants, and the environment. Here in Vancouver, Genome BC has been leading the way in the expansion of knowledge and specific beneficial advances in science and technology. COVID-19 research was one such benefactor. I invited Pascal Spothelfer, the about-to-retire CEO of Genome BC, to join me for a conversation that matters about the role genomics is playing in our lives and in the expansion of scientific research and biomedical technology here in BC. Pascal, welcome.
Thank you, Stu. Thanks for having me.
How important is Genome BC in the sphere of political research or scientific research, maybe even political research in British Columbia to begin with, and then what's our role nationally?
Genome BC has a pretty prominent role in BC when it comes to research and innovation. Generally speaking, research is mostly the funding kind of domain of the federal government, less of the provincial governments. However, different provinces have different setups on how they fund research. And in BC, Genome BC is one of the main recipients of provincial funding to then fund specific innovation in genomics or omics in general, together with probably the Michael Smith Health Research, who is another important health-focused research organization here in the province.
So how does it work? Let's say we have biomedical research that is happening out at UBC, and they then say, okay, we're working on this particular enzyme or this component of human health. We need help from genomics or genomics research. What does that relationship look like, and why is it important that there be this symbiosis?
Yeah, you asked the national question as well. So Canada was behind other nations when the human genome was sequenced in the late 90s, and then the federal government, on the urging of scientists, established Genome Canada, a specific funding agency to fund genomic research. And as opposed to the traditional funding agencies in Ottawa, it was done in collaboration with provinces who in turn put up these genome centers, one of them being Genome BC. So how does it work? Genome Canada would put forward a competition with a certain theme for research, and we would then reach out to our regional researchers and work with them to put forward competitive proposals to Genome Canada that would then decide in a peer-reviewed process which projects get funded. The ones that get selected out of BC, we would then co-fund, so we'd provide additional funding to the researchers, and that would then make up a research project that would last for 3, 4, 5 years. And there's different programs that are available. Some are more research-oriented, some are more translational, have a more translational orientation involving industry, etc. So that's kind of the federal layer. And the same researchers are also putting proposals forward to other funding agencies. But the genomics funds would look to us to provide them with the necessary funds.
So how important then is that relationship if somebody is in British Columbia and they're wanting to tap into that national funding? What's the likelihood of them being able to get the same kind of exposure if it weren't for this partnership with Genome BC?
Well, it would be more challenging for them because we also work with them on the development of the proposal. We prepare them for the peer-reviewed process that includes panel presentations and other things. And more importantly, we are with them through the delivery of the project, both scientifically and financially, and we are with them between projects. So we have a very good understanding of how the ecosystem look like, and when there are opportunities that arise, we also know whom we would go to and ask and talk to develop funding programs.
Can you give me an example of a few projects that have been funded and have worked their way through in the last few years?
There's a whole range of programs, but let me go back to a competition that was launched in 2017, a large-scale applied research program around health and precision medicine. And we did very well in this competition. And so let me give you one project that was operating out of the BC Children's Hospital. And it was around pharmacogenomics. So essentially using genomics to understand the child's genome and find out whether that child would be susceptible to an adverse reaction to a specific cancer treatment. So Dr. Bruce Carlton was the principal investigator, and the problem he was combating was that certain cancer drug, one specifically, led to deafness in children that were treated with that drug. So by using that test that he developed, this could be avoided and another drug would be administered to that child. Enormous impact for these children that are affected. And that was a 4-year project that was co-funded between Genome Canada and us and was successfully concluded. And that application is now being used within BC Children's Hospital and nationally, thanks to Bruce's efforts.
Well, that really is, I think, the great hope of most people when they think about genomics, that with pharmacogenomics, you're going to be able to give me a bespoke treatment. How close are we, though, to it actually finding its way into the average clinician's practice?
We're quite a bit away from the average clinician's practice. And there is different applications. So for instance, there is a set of about 30 or 40 drugs where there are established tests known that you can do. So for instance, one is a blood thinner warfarin, and some people react very quickly to it and others very slowly. Kind of important to know, because if you react very quickly, you might want to give 1 dose and wait. If you react very slowly, you want to give 1 dose and wait a bit longer. Because you don't want to say, oh, there's no reaction, I give them more. Mm-hmm. And so that's something that could actually be rolled out, but illustrates one of the challenges. When you are a general practitioner in British Columbia, where do you find the time to administer that test? It's not reimbursable now. How do you understand that test as a physician? It wasn't part of their education in the first place. And even if you do the test, you then have to know how to use it and collect the data that's necessary so that patients can actually benefit from that application. So that shows the challenge of implementing new solutions in the healthcare system. However, on the tertiary care level, so if you go into BC Cancer and you're being treated by an oncologist there, you have access to some of these genomic technologies today.
So we're already starting to realize the benefit of that.
Yes.
How long do you think it will be before we're able to expand it out beyond just, tertiary care facilities?
As so often, it depends.
Yeah.
Certain things are easier to roll out than others, and generally takes way longer to roll these things out. But that's also when an organization like Genome BC comes into play,
right.
Because when somebody manages a healthcare system, it's all about patient impact, standard of care, and affordability of the whole solution. And I think what we can do as an organization, we can work with the Ministry of Health and healthcare delivery organizations, and we can fund projects, pilot projects, where we test these things following certain parameters. So we could show how much money can be saved by applying a pharmacogenomic test in the primary care system. It takes a while to do that, but we would be the organization who then works with the Ministry of Health to putting that in place, collecting the data, and then show the evidence that could help them to make a decision in that direction.
So we're moving in that direction, but it's going to take time. What I find also interesting about the ability to now decode the genomics of so many different elements of life is that it can help us make a wide variety of other decisions. agriculture, which I believe is a very important one. What role does Genome BC play in helping the agricultural sector?
We are increasingly working with that sector, and again, it's what is the capacity in that sector to actually use solutions that have been developed. Over the past few years, more and more researchers are paying a lot of attention to it, which is the basis of it all. If we don't have the research, there's really nothing we can do. And now we come to the point where selective breeding of trees, for instance, is now common practice. In BC, pretty much every tree that's being bred, they use genomics to select the traits that they want to get.
In trees?
In trees, which allows you to make decisions on what trees to plant today, which will be climate resistant in 40 years when they have matured. And but there is other areas, be it in wine, what kind of yeast to use, be it in blueberries, what kind of strains to use. There is research that is done on sunflower, etc. And all this research is flowing out in the community, and which leads to the next challenge, which is the acceptability by society of food that has used genomics as a tool to be grown and to be selected.
Well, that is a big challenge because there is an anti-GMO, which is in essence what we're talking about here, movement that says, well, if you're going to mess with the genome of that plant or vegetable, somehow you've made it that it's not whole.
Well, that's not what we're talking about here.
No,
actually.
I know.
No, that's not what we're talking about.
Yeah.
So genomics can be used to identify certain traits and then breed accordingly. So instead of growing a tree and waiting 40 years and test whether it withstands the weather or not, we can look for traits in trees in California and others and see whether we find similar traits in our trees here and then breed specifically the ones that have it, which is not the same than modifying the genome, not at all. It's just a selection of the right genome that is out there. And there has been a lot of discussion around GMO, and it has gone through multiple iterations. It started really with scientists bombarding genomes with X-rays and see what happens. And today we have much more sophisticated tools that allow us to do things that have far less that are very different from what was done when the GMO movement was created.
I talked to a scientist from UC Davis, I believe, who was working on trying to increase the survivability of rice underwater from 3 weeks to 5. One small little change. But the work is incredibly complex, but if successful, could increase rice yields by the billions of tons. This is the kind of work that can make a huge difference in the quality of people's lives.
It can make an inordinate difference in the nutrition of the world, and we are not able to feed the world in the years to come, in the growing population, without yielding much more food out of the growing the land that we have, and genomics will play a major role in that. And I think where then the next discussion comes from, to do that in a way without disrupting the economics of agriculture. And that's one of the problems that needs to be addressed. Because as soon as seeds are genetically modified, now how are they being sold? How are they being marketed? What is the dependency of the farmer on the seed producer? These are all questions that are not genomic questions. They're legal, ethical, economic questions, but these need to be addressed. As with any other leading edge technology, it's important to have this parallel research and understanding of the natural science and the social science and the humanities.
Right. I talked to or interviewed a crop scientist, and I was asking him, how do you determine how you're going to make a change in a kernel of wheat if you're growing it in Idaho versus Saskatchewan? And he said, oh no, it's far more specific than that. It's whether you're in this county or on that side of the hill. Like, we're getting to this point because the whole idea is to be able to increase yields. So that's dealing with the plant. Are you also looking at the natural genome of the soil?
Absolutely. I think the soil genomics is becoming increasingly important. It has been recognized in its importance when you talk about forestry as well, but certainly in agriculture. And environmental DNA is a scientific discipline within genomics that's incredibly important, because it allows to take relatively small samples and to identify the inhabitants, the living creatures within that soil. I mean, the most practical example we see right now of environmental DNA is the wastewater samples that are being taken to see what is the incidence of COVID-19 in a certain population. And that's by taking water samples, finding DNA that belongs to the COVID-19 virus, and then determining what is the load, the viral load within a certain population. And the same kind of technology is then applied to soil, for other reasons in water and other substances.
Well, I can't help but think about marine biology as well.
Yes.
Very, very important, especially here along the coast. Are you working in projects that are addressing some of the issues that are perplexing at the moment?
Yes, and I think we live in a province where agriculture is a hugely difficult issue to deal with, because on the one hand, it's our largest export article, and on the other hand, it has big consequences for our natural habitat, for indigenous people on the coast. So this is a big discussion. Having said that, we just had a conference of genomics forums a few weeks ago, and in a panel around fisheries and aquaculture, or the blue economy, one of the participants said, so the Earth is covered 70% with water. We produce 2% of our entire food from the water. So if there isn't potential there, I don't know where we could find potential. But it has to be done right.
Mm-hmm. So you mentioned COVID-19 a moment ago. How important was the relationship between the research that was happening here in British Columbia, which was extraordinary, played a very important role, and that with Genome BC?
You know, it's interesting you ask, because we had our AGM last Friday, and we obviously have a luncheon, a community kind of a luncheon of our ecosystem. And I talked with two colleagues of the BC Centre for Disease Control, and they mentioned to me that they couldn't overemphasize how important it was that they had a close relationship with Genome BC, particularly at the outbreak of COVID-19, because they have their budgets, they have their work processes, etc. Now they suddenly had to shift. And they needed money to sequence the virus as quickly as they could, and not only sequence the virus, but to put a pipeline in place so they could do it over and over again. And we provided them with $150,000 essentially in a few days, which got them started. They've leveraged that money over and over and over and over. But then we continued to work with them, and whenever they saw gaps, we could actually step in and help them to fill these gaps. And today they have a process in place, they have instrumentation in place, they have the training in place and the procedures in place, so they can take a virus, put it into their system, and they can recognize very quickly what is going on. So should new pathogen emerge, monkeypox is in the press right now, the same kind of procedures can be applied by the BC Centre for Disease Control, which was built on this cooperation during in the early days of COVID.
So an issue that I think is very important in British Columbia that doesn't get the kind of attention that I'd like it to have is just how important is the biomedical and life sciences community. And we need to be able to create opportunities to attract talent, grow that talent, and then help to support those organizations that will benefit from it. What role does Genome BC play there?
We fund research and we fund innovation, and we do it in a number of ways. So if you look at the large competitions, generally it's the senior investigators that will get the funding, with all the diversity issues that is being created by that. Because the people who have done research in the past are generally the ones that attract more funding. So we have an opportunity as an organization, we have done that. So we have opened smaller competitions to start with for young, up-and-comer principal investigators. And suddenly what we saw, the percentage of females became much larger, the percentage of diverse ethnicities became much larger. So we could track, we can see that. And it is by engaging broadly and bringing up that talent broadly that we can make a real difference.
Do we have the potential to be a global center of excellence in research and scientific advancement in life sciences and biotech, or are we still a contributor?
Well, we are the global leader in lipid nanoparticles, which is a tiny but very important part of mRNA delivery. We're global in that. Are we global in other aspects of biotech that are important, say antimicrobial resistance? We may not be. But I don't think that this really matters, whether we are a global powerhouse or not. What really matters is that we can build and we can go from one strength to the other. We can grow it. It can become more important. It provides more jobs. It provides more solutions that also find their way in our healthcare system.
You are about to move on to other adventures in your life. From your time at the helm at Genome BC, what are some of the elements of your experience that you go, man, I learned a lot, and here's what I am most proud of helping to make happen?
Yeah, I certainly learned a lot about how deep our system is and how cooperative the system here in British Columbia is. And the depth, it's not just Vancouver, there is incredible ingenuity across this province. So I also learned that we are way too slow to adapt these solutions. We have to learn to become more aggressive, take bigger risks, and adopt some of these things that we develop here and adopt it here and make it work here. And from a proudest moment, I think certainly the ability that we had to respond to COVID-19 was a very important moment for the organization. But then I think at the end of the day, we've been now around for 22 years. And I still think we are extremely relevant and remain relevant. And that's probably the most important thing for an outgoing CEO to be able to say.
Well, as an outsider, I agree. I think that its relevance continues to grow because it plays such an important part in the ecosphere of scientific research and scientific community. Well, I wish you great luck as we move forward.
Thank you very much.
Yeah, thanks for coming.
My pleasure.